Can everyone hear me? Okay. Hi, everyone. My name is Anna. I'm a PhD student in Graham Thompson's lab at Western University. Thanks to the thanks to the organizers of BeeCon. It's always nice to be here. And the title of my talk is The Brain-Gut Axis of Honeybees, How Microbiota Affect Individual and Collective Behavior. So the brain-gut axis is commonly applied to the human condition because according to the Holobiont theory of evolution, the individual human or otherwise is deeply symbiotic with gut microbes. And those microbes in turn can influence host metabolism, elements of immune system, and behavior through the signals that they pass from the gut to the brain. The brain-gut axis is therefore indicative of individual's health and ability to function. And this makes sense because the gut is, after all, the first point of internal contact with the outside environment. And it's has a function of processing nutrients and food, as well as producing essential amino acids. And what's interesting about this brain-gut axis is that there is a potential to study it at different levels of biological organization. For example, for highly eusocial taxa like honeybees and ants, where selfless individuals direct their behavior towards others, there's a potential to study the brain-gut axis at the collective behavioral level. So here I'm using the Western honeybee as a model to study how gut microbiota can influence individual and social behavior. As trendy as the brain-gut axis research has recently become, for the most part, it concentrated on humans and or vertebrate models, and not as much has been done for insects, and even less has been done for eusocial insects. And as you can see from my cartoon here, I have highlighted the brain of the bee in blue and the hindgut in yellow. And the hindgut is considered to be a site with richest microbiota in honeybee because this is where nutrient extraction usually occurs. So presumably perturbation of the hindgut microbiota by applying either antibiotics or probiotics, as you can see in the image, here could potentially really influence behavior of individual bees, but also, as in my hypothesis, it can influence collective behavior of entire colonies. So just as individual worker bee influences the queen, other worker, and larva, perturbation of the brain-gut axis via antibiotics or probiotics could amplify its effects and spread out across the entire colony. So in the slides that follow, I measure how the brain-gut axis could potentially affect these four behaviors: foraging, defense, hygiene, and recruitment. And as we know, bees don't forage for themselves. They do so on behalf of entire colony. And so these behaviors that I am measuring are truly social. But first, before measuring these social behaviors, we needed to choose the probiotic strains and come up with a method which allows us to effectively deliver probiotics to the hives. So there's not much precedence for it, but we developed a novel way of delivering antibiotics and probiotics into the hives via what we call this biopatty that you see in this image. So beekeepers, in the springtime, beekeepers generally use pollen patties as supplements to help start their colonies. So we simply modified this delivery method by putting either probiotics or antibiotics into these patties, which we then call biopatty. So one of the most interesting or important aspects of producing a biopody is selection of a strain. And so we do know that honeybees are lactobacilli rich. And so some of our strains use that bacteria. Okay. So once we make these patties, we deliver them to the hives, and we recently just finished our field experiment, which looks like this. So we had 4 treatment groups. We had a probiotic patty group, antibiotic patty group, no patty control, and then we had a vehicle patty. And the vehicle patty is, again, what beekeepers normally use. So it just has the pollen, but it just has the protein, but it does not contain probiotic or antibiotic supplements. And we did this in a randomized block design for a total of 20 different colonies. And you can see in the picture below, this is how we deliver these biopatties into the hives. Okay, so here we go with the first of our assays. Here we ask, how do perturbation in the brain-gut axis affect social foraging? Here we're using a simple assay, and we're measuring the number of foragers that are exiting and entering the hive. And you can see the setup here. It's on the ground. You can see the white Bristol board. This is simply serves as a good contrast, and there is a video camera mounted on top of the hive. And this allows us to see how many foragers leave and re-enter the hive. And the Bristol board on top simply acts as a sunblock to prevent from overheating of the cameras. Okay, so here's a snippet, 10-second snippet of the video. You can see a pretty nice contrast against the white background, and you can see foragers. And then on top of scoring the net foraging effort, we also measured patty consumption, which you can see in the middle photo. You can see that the honeybees have pretty good appetite for the biopatties that we produce. And we also estimated the amount of pollen that was being collected using our really awesome pollen traps. And you can see that these are pollen grains that are being scraped from the foragers as they return into the hive. Okay, and this is the second behavior that we measured by asking the perturbations in the brain-gut axis affect social defense. So here we provoke defensiveness using this chemical, which is called IPA. IPA stands for isopentyl acetate, and it's a chemical that basically mimics the famous banana smell that is produced by the hives whenever there is an intruder. So here you can see, this is my supervisor who volunteered for the video. You can see them pretty aggressive here. So here you can see Graham waving the flag to stimulate the defensive behavior. And so more formally, you can see in the center photo, this is the resulting plug after one of the assays. And you can see me there in the picture staying away from the hives, safely counting the stingers. And this is just the top view of the assay. You can see this was taken on August 11th this year. Because we also recorded a video so that we can use it to, we can analyze it and estimate defensiveness. So we were measuring this twice a week over a 4-week period. And so here, this is a preliminary analysis of a defense. And on the x-axis, you can see the 4 different treatment groups, the antibiotic, control, probiotic, and vehicle groups. And on the y-axis, you see average number of stingers per sampling day. So here we can see that the vehicle supplemented treatment group, which is seems to be the most defensive, and the probiotic and antibiotic supplemented groups appear to be most similar to one another. So besides measuring the behaviors of the honeybees in the field, I want to bridge the gap between the gut and the brain by using more mechanistic approach. So for this, for each day of our assays, I've collected samples of live bees and put them into liquid nitrogen. So from those samples, I am going to dissect their guts and their brains. And I'm going to measure the absolute and relative diversity and abundance of microbes using 16S RNA amplicon sequencing, as well as we plan to use targeted proteomics and metabolomics. For the brain dissections, we are looking at specific regions such as subsphagial ganglion, which is thought to be responsible for spatial learning, as well as mushroom bodies and canyon cells. Okay, so to summarize, bridging the gap between the gut and the brain requires careful consideration, especially when selecting different strains, so that it would be possible to track down the scaling effect from the gut to the brain to their behavior. And so far in our work, we were able to develop a novel method of probiotic and antibiotic delivery to the hives. And we showed that vehicle-fed colonies appear to be more defensive. And for our next season, we plan to also test their hygienic and recruitment behaviors. And with that, I would like to thank my lab and you for listening. And I'll take some questions. Yes. No.
That's really great talk. I'm just curious, have you figured out a protocol for
it's very small, yes. Oh, okay. So the question was whether or not there has been a method developed for dissecting the brains. This is something we're still figuring out. I do understand that the regions are small, so perhaps we plan to use different types of staining. Maybe not necessarily dissecting it, but just measuring the regions. That's a good idea. Yeah, something. Yeah. Yes. This is something that, yeah, is their equivalent of the blood-brain barrier. So this is something that I think is happening. I think that the gut is not directly connected just to the brain. It's probably occurs through the blood barrier. And if you look at the anatomy of the honeybee, there's actually a connection near the hindgut. There's a connection that I think maybe that's the region that should be looked at. Yeah, but I think so. Sorry, repeat that again. I was gonna repeat it, but one of the things that we also want to look at is potentially collecting hemolymph to see if we can track down like metabolites that are produced, by the gut and whether they go into the brain or have effect on the brain. Okay.
Thank you very much. In the interest of time, we'll cut that off there.